As Cliff said, my name's Greg Tylka. I'm from Iowa State. I have the luxury of being able to focus almost exclusively on soybean cyst nematode. I've been at Iowa State since February 12th of 1990. So it's been a long journey. It's been a fun journey. And this is the most fun part of the job, is interacting with folks who work with farmers. Normally I would encourage questions, but we've decided we're not going to do that until the end, is after I've gone through the materials. But at least maybe show me that you're paying attention, maybe bug your eyes out or blink or something. Try not to drool as you're falling asleep to show me you're engaged. And I'm going to walk around some as well. Last thing I'll warn you is if it looks like I'm in pain, I am in pain. I pinched a nerve this weekend, and so it hurts to stand, walk, or sit. So I don't know what other options there are other than to lay down on the table here in front and talk to you. So I'm going to be moving around, and if I sit down, it's not because I'm lazy, it's because I'm kind of hurting. So with that, am I forgetting anything, anyone? I'd normally also ask people like Sean to interject his perspective, but I think we're going to just have to hear about that after we're done with my presentation, then we'll move on to Sean. So, okay, so soybean cyst nematode. It is a worm that lives in the soil. There are lots of other nematodes that live in the soil as well that are beneficial to agriculture and soil health, but soybean cyst nematode obviously is not, and that's why we want to control it. Like all nematodes, soybean cyst nematode starts out as a tiny microscopic egg, and what hatches out of that egg is a worm that we call a juvenile. And that is a weak link in the life cycle that I want you to remember, put in the back of your head. This little worm, once it's hatched out of the egg, it's vulnerable to being eaten by other things that live in the soil or to starvation. So it really needs to get into the safety of a soybean root to be protected and to be able to develop through the rest of its life. And when it gets into that soybean root, it burrows to the center of the root where the vascular tissue is. And once it attaches to the vascular tissue, it starts to feed and swell. And as you can see in this bottom photograph, the swollen juvenile, it is swollen. It no longer looks worm-shaped. That's because it has fed for several days. Now at this point, I can't tell you if that nematode in the root, in the bottom image, is a male or a female. They look identical. But this is as large or as swollen as a male is going to get. And if it's going to become an adult male, it gets this large and then it reverts back to a worm shape, believe it or not, and it leaves the root and goes out into the soil. So in the photograph on the right, we see an adult male. It's a long, thin worm, kind of like a large version of a juvenile. I should point out also that these aren't all at the same scale microscopically. So this is much larger than the swollen juvenile, the adult male is. So if it's an adult male, it does the first three images, swells to a swollen juvenile, and then reverts to a worm and leaves the root. If it's going to become an adult female, it continues to enlarge until it takes on this lemon shape that you see to the left in the photograph on the right, the image on the right, adult female. And so we often call it lemon-shaped. So these things, though, stay attached to the root and feed throughout their life cycle. So the adult female is attached to the root. The adult male is out into the soil. The female is so large that she actually ruptures out of the root and is exposed on the surface of the root. And she sends out a chemical signal to attract males, and they mate. And a female will be mated by many different males, and males will mate with many different females. So there's a lot of genetic mixing going on. After the female is mated, she'll produce eggs. She makes about 50 eggs outside of her body in a clump called an egg mass, and then she can fill up with another 250 or more eggs internally. So total, there'll be 300 or more eggs produced by a single female, and then she dies. And it's the body wall of the adult female that becomes brown and tough and leathery and hardens off that forms the cyst. So the middle name in soybean cyst nematode, the cyst, is a swollen, dead female full of eggs. And the circle of life then continues. Couple of other last points about this. Once we get to the middle of summer and the soils are nice and warm, the nematode is able to complete a life cycle turn or generation in about 24 days. And so if soybeans are growing in a field for 4 months, 120 days, we can get many different turns of the life cycle, many generations. Generically, I just say 3 to 6 generations. So the coolest thing, though, is that bottom image that I just showed you. And I want to take a closer look at it. If you're a biology nerd like I am, this is just fascinating. So here's a close-up of that image. So here's the outer edge. The top shows the outer edge of the root. You see the vascular tissue labeled in the center of the root. You see the swollen SCN juvenile in an area of the root called the root cortex. And then the cool part is this dark area around the head of the nematode called the nematode feeding site. This feeding site is, the technical name is a syncytium, and it is formed by it starts out with one living plant cell, and then it merges with adjacent living root cells to form one big cell called a syncytium or feeding site. And the amazing thing is when it becomes a syncytium, it looks like no other cells in the soybean plant. So the nematode goes in and changes a single plant cell to recruit cells from around it. They all merge together and they form something new that doesn't exist anywhere else in that soybean plant. And that cell, or syncytium, has extremely high metabolism, presumably to feed the nematode. And that whole process of forming the syncytium is caused by something that the nematode is producing and injecting into the plant cell. And that something is produced by the juvenile. Remember the worm that hatched out of the egg. It has a mouth spear called a stylet that you can see in the top left of this image labeled stylet, and it punctures a plant cell. It's hollow, so it can inject stuff out of the nematode's body into the plant cell, and then it can bring back food through that same spear object. And we believe something in the glands, the subventral glands, or there's another gland called a dorsal gland, is being produced and injected into the plant cell to change it into a feeding site. So it's pretty amazing, and it's one of the mysteries currently of the biology of soybean cyst nematode. So the biology of soybean cyst nematode makes it difficult to manage in 2 respects. One is how quickly it builds up, and secondly, how well it survives. So this slide is talking about how quickly numbers can build up, and we've kind of touched on this object. You should now identify or be able to think of it as a cyst. It's a dead female that's died, and it's kind of brown, and you can kind of see eggs inside, and there's some eggs outside of the body. In a study published in the 1980s, they came up with about an average of 300 eggs produced per female, combined between the egg mass and inside the dead cyst. But in nature, in soil, that number is really about 250, we've learned. For some reason, the nematodes just aren't as vigorous growing in the soil as they are in a laboratory, which is where this study was done. But I'm into numbers. I love data. I love numbers. And does anybody here watch the TV show Big Bang Theory? And they're constantly saying, do you want to do the math? So I want to do some math here with the numbers. So I'm proposing that we start out with 100 SCN eggs, and I want to do math over 6 generations, okay? So here's how we do the math. We take 100, and the first thing, if we want to figure out what that results in, we have to divide by 2. Does anybody know why we have to divide by 2? Males and females, good group. So only half of that 100 are going to become females. So that automatically makes that 100 50. But then that 50 females can produce 250 eggs each, and that gives us, just after 1 generation, 12,500 eggs, okay? And then if you take that 12,500 eggs and do this all again, half becoming females, 6,250, 250 eggs per female, in 2 turns of the life cycle, 1.56 million eggs, okay? And here now is what the whole math looks like. I stopped after 3 generations. My calculator doesn't have more than 9 digits. And so you get a feel for numerically how quickly these numbers multiply, okay? But I can guess what you're thinking, and that is, well, this is crazy. That doesn't happen in nature. And you're right, it doesn't happen in nature. So I want to introduce the concept of attrition. So attrition is just the natural decrease in numbers that happens in nature. One of the textbook definitions is a reduction or decrease in numbers, size, or strength. And there's attrition in soybean cyst nematode as it develops. And so we can now start doing math and figuring out, well, what do you think's the attrition rate? And when I ask groups what the attrition rate is, I hear guesses of 50 or 60 or 70. And I'll tell you, those seem reasonable, but they're extremely low. In some other nematode literature, it's up in the 90s, so I just picked 97% to work with. So assuming that there's a 97% attrition rate, I want to do the math again. So we start out with 100 eggs, and we end up with 12,500 after that first turn or first generation. But then attrition is going to kick in. So this time, from that 12,500 eggs, we're going to only take 3% of those because we're going to assume 97% attrition. 97% die off before becoming adults is what this means. So that brings us down to 375 individuals, of which only half are female, 187, multiplied by 250 eggs per female. We're still at 46,750 eggs after the second generation. And here is how those numbers play out with 97% attrition. From 100 eggs to 9.2 million. So we know that's not true because even those numbers are crazy high. So let's look at 98% attrition. At this point, I don't have to do more of the math for you. It's the same setup, but I'm now taking only 2% of the population. This is what the numbers play out to be. And then if that's still too high, which I think it is still too high, finally, 99% attrition. So even with 99% of the population not making it to adulthood, 100 eggs can become 37,000 eggs in 6 generations. So that should give you some feel for the reproductive capability of soybean cyst nematode. And if you're wondering how high egg counts can get, I took a photograph of this as I was leaving, right before I was leaving yesterday. This is in my lab. And we have listed, to our record, since 1990, the highest egg count we've ever made in a sample was 334,800. So crazy things can happen. I mean, not into the millions, but that was a real sample from a farmer. So hopefully I've convinced you that the nematode has high reproductive capabilities. The other thing we talk about is it survives extremely well in the soil. And unfortunately, there aren't a lot of good numerical data to back this up, but we have to rely on some of our forefathers in the realm of soybean cyst nematode. And someone I consider the grandfather of soybean cyst in the United States is a fellow named Bob Riggs from the University of Arkansas. And he writes a passage in a book chapter that he wrote, shown here at the bottom, that says that he has experienced being able to recover viable eggs from SCN cysts from a field that only had fescue, a non-host grass, grown for 40 years. He was able to recover live eggs in cysts. And then another contemporary of Bob Riggs was a fellow named Darrell Slack, and he told a story of where they had an SCN-infested field that they kept free of plants, both planted plants and weed-free, for 8 years. They grew susceptible soybeans for 2 years after that, and by year 10, those susceptible soybeans were being damaged by soybean cyst nematode. So in general, we say that soybean cyst nematode can probably live in the soil 10 years or more. Well, soybean cyst nematode was first discovered in North America, in North Carolina, along a coastal region. And then it very quickly spread. The animated map you're seeing right now shows it spread from that area of North Carolina where it was first found, a little bit through the south, and then it makes a big jump over to the Boot Heel of Missouri, and it seems to be spreading up and down, or have spread up and down the Mississippi River. And then it makes another big jump up to north central United States in the late 1970s, and it seems to be spreading from there as well. And so it's rapidly disseminated across the soybean growing areas of the United States. Well, at Iowa State, we are currently the keeper of the national map of where soybean cyst nematode has been found. And every few years we publish that map. Here's what the map looked like in 2014 when we published it. And during that year, then we also went to the USDA and tracked down where soybeans were grown in the United States. And I've underlaid the soybean producing areas or counties of the United States in blue. So obviously any area covered in red where soybean cyst was found, there were soybeans grown. But then the blue showing here in this map show soybean producing counties where SCN had not been found as of 2013, 2014. But soybean cyst has covered a good 70–80% of where soybeans are grown in the United States. And then here's the most recent map, and I'm glad I included Canada because I know we have someone here from Canada as well. This map was published just earlier this year by our group, and this little box here to the right shows the new counties that had been discovered since 2014, since the last map. And the notable ones there are Iowa, you see, had added one new county. And the reason that's notable is it puts us as only the second member of the club of states that have SCN in every county. Illinois is the only other state that can brag about that or complain about that. We're looking to recruit Indiana at some point in the future. And I don't know how many other states will be part of that club. Another notable one, probably the most notable thing, is New York. There are a fair bit of soybeans grown in the state of New York, and SCN was found for the first time in one of the counties in New York. And then the numerical winner or loser of the competition is North Dakota, when they had added 7 new SCN counties in that 3-year period between 2014 and 2017. So this is where we're at currently in terms of North America. So these maps are fine and dandy, and they're kind of pretty to look at, but one field found in a county puts that county on the map, and that's exactly what happened in this particular case for Iowa. We found soybean cyst nematode in one field in Allamakee County, and it put it on the map. But it really doesn't tell you how widespread the nematode is in a particular area. To do that, you need to do some kind of intensive sampling and surveying. And we hooked up with the USDA National Ag Statistics Service, who randomly visit fields every year to measure corn and soybean growth. That's how they predict corn and soybean yields. Well, in the mid-90s, we paid those folks to pull a soil sample for us. And all that soil samples were sent to Iowa State University. So we had something like 1,400 randomly selected fields sampled by the USDA NAS enumerators, they're called, and all came to Iowa State. We tested them for SCN, and here are the percentage of fields in each state that we found soybean cyst nematode. Much higher than we had expected. 75% in Iowa. Illinois always has to outcompete us, and they're at 83%. And then we see the percentages in the other states involved in that survey. One final thing I'll tell you about this is, obviously this is old data from the mid-90s. I did a random survey of just Iowa with the same USDA folks in the mid-2000s, and we came in at 72%. And then Terry Niblack, when she was at the University of Illinois, she redid the Illinois survey with USDA NAS people in the decade of the 2000s and came at 82%. So I thought that was really striking that 2 of the states had done random surveys 10 years later and came up with roughly the same percentages. And then the last point I'll tell you is we're redoing the Iowa survey again right now, this year and in 2018. And we have half the samples from 2017 in Iowa processed. And you want to guess what the percentage is? It's at 70% right now of the samples we've received so far. So I'm really curious to see if Iowa stays around 70 to 75% of the fields have SCN over 3 random surveys conducted over 20 years. But it is obviously very widespread. It's so widespread and the damage potential is so great that it is, in almost everybody's opinion, the most damaging soybean pathogen in the United States. And There are a group of plant pathologists that don't include me, so it's not like I'm swaying the results, that rank the most important soybean pathogens and actually come up with yield loss estimates. And they've published a recent report. It was published in 2017. It represents the yield loss that occurred from 2010 to 2014. Tom Allen and 40 some other co-authors on the paper published it in this journal online. And soybean cyst was number 1, and we see the others. I list the top 10 there. And then worldwide, this same group led by another person, Alan Rather, who has retired now but used to be at University of Missouri, ranked and came up with yield loss estimates for the world, soybean pathogens, and soybean cyst nematode was number 2. Number 1 worldwide was soybean rust. But soybean rust really hasn't had the major impact in the United States that we had expected. But soybean cyst nematode is certainly number 1 in the minds of many soybean plant pathologists. And it's because of images like this. Here's an aerial photograph I took. This is probably a 20-year-old photograph from central Iowa showing severe damage. And in situations like that, if you grew a resistant and a susceptible soybean, you might get double the yield out of a resistant variety than a susceptible. So when people ask what's yield loss, what's a percent yield loss that's caused by SCN, I immediately think of ranges, first off, because it's not a single number. And one number I think of is 50%. To me, the data shown on this slide show 50% yield loss. If you didn't grow a resistant variety, you got half the yield of a resistant variety. Now, it's probably more than 50% because that resistant variety isn't yielding as it would have if the nematode wasn't present. We know that even resistant varieties suffer some yield loss, but it certainly yielded double of the susceptible. Now, one of our big problems in the Midwest is this is what SCN-infested fields look like. Beautiful, lush, green plants. And in this scenario, these are what yields look like very often, where you might only get a 5 bushel per acre yield difference between a resistant and a susceptible. And some might say, well, I mean, that's hardly worth thinking about, but it's 10%. Even this little difference, that visually it looks little in terms of a bar graph, it's a 10% yield loss. So soybean cyst nematode is extracting a toll, and how bad of a situation it is dictated by not only the nematode, but weather conditions and the soybean variety. Plant pathologists wrap these 3 factors into something they call the disease triangle. If you've ever taken a plant pathology class, this is like day 1, 15 minutes into the course. It says that the amount of a disease that occurs depends on how aggressive the pathogen is, how susceptible or vulnerable the host is, and how conducive the conditions are. And that whole mindset or concept works really well for the yield loss model with soybean cyst nematode. There is a certain set of conditions that favor SCN reproduction and yield loss, and it's 2 simple words, well 3 with the conjunction. It's hot and dry. Soybean cyst nematode goes crazy and yield loss is extreme in hot and dry conditions. But I don't want to mislead you. Whether you're dealing with a situation shown on the left or on the right, underground, soybean cyst nematodes churning away. And it's building up numbers. And something that's fairly intuitive that for people that are data-driven is the higher the number, the greater the yield loss. So how would you know if you have soybean cyst nematode when fields look like this? It's really a psychological challenge to convince somebody to think about looking for what's going wrong in this field when the beans look so good. Well, there are 2 ways to scout for soybean cyst nematode, and neither one of them involve driving in an air-conditioned pickup truck at 75 miles an hour, unfortunately. So you have to stop the pickup truck and do one of 2 things. You can dig roots and look for females on the roots or collect soil samples. Both are really straightforward. Digging roots and looking for females, the most important word in this slide is the first word, dig, as opposed to pull roots. You need to take a trowel or a shovel and pop out a clump of plants. You can do it as early as 5 or 6 weeks after planting through the month of August for sure, or at least through the month of July into early or mid-August. The problem is we get later into the season, the nematode females are forming on young roots, and in August those roots are 3 feet deep and hard to get at when you dig with a shovel. So kind of 6 weeks after planting through the month of July is prime time to do this. And really, you just dig roots, shake the soil off of it, and look for the little white females. The advantages of this method of scouting are 2 of my favorite words, and that is quick and cheap. It's free, in fact. You don't have to pay anybody to look yourself. There are limitations in that you can only do it, as we've just discussed, kind of 6 weeks after planting through the month of July, maybe into early August. And then secondly, you don't get quantitative information. So in this photograph here, we see 4 or 5 females. There might be another female hiding behind a clump of soil there. But that could lead you to think maybe the numbers are relatively low, but you could come back a week later and see literally 10 times as many females on this same root. Because they kind of pop out in cycles, because the life cycles kind of cycle through the season. Of course, the other way, as I said, to check a field is to collect a soil sample. That is really easy to do as well. The only thing you got to remember is put the pointy end of the probe in the soil and push. So it's great in that you can do it any time of the year and you get quantitative information, although you see I have the word quantitative in quotation marks. We'll talk about that in a minute. The limitations are you have to pay somebody to process that sample. And then secondly, you've got to wait for the results to come in. But it's perfectly fine to do this as a way of scouting as well. So how would you sample? It's pretty straightforward. We need soil cores. We'd like 20 or more, actually. We're always asking for more and more soil cores to a depth of 8 inches. And you want to collect that from many different places in the field. You use a zigzag pattern or an M pattern through the area. And we'd like 1 20 or more core sample for every 20 acres or so. So that's a pretty intensive set of cores or samples in some respects. But it's because the nematode is so aggregated in the field. So the way you could do that, here's an example. This is how I view the world. I'm a bit obsessive compulsive and I like everything nice and neat in squares. And so if I had an 80-acre field, I'd divide it into 4 20-acre parcels, and then I would zigzag my way through and collect a 20-core sample from each one of those. But there are more reasonable ways to approach it. Probably everybody in this room has way more common sense than me and realizes that a field isn't uniform in quadrants, and so they would view the field in terms of zones, management zones or areas, and might take 4 separate samples depending on some agronomic feature of that field, and that is wonderfully fine as well. And then another approach would be to really target your efforts. And we know there are some high-risk areas to first have soybean cyst nematode in the field, and those are illustrated in this diagram. And so you could collect your soil cores from a field entryway where soil could fall off of farm implements as they're moving from one field to the next. We know soybean cyst loves high pH, and so high pH area of a field you might sample. An area of a field that you just can't figure out why the yield is so low. Low spots and previously flooded areas where surface water might move soil from surrounding areas. And then along a fence line is kind of a quirky place, but I've seen it at least once in my career where I saw a strip of yellow plants that had soybean cyst nematode along a fence line. And we hypothesized that wind-blown soil kind of hits that fence line and then falls down right there near the fence line. And so some believe that might be another place where you first find soybean cyst nematode. But ultimately, it's about taking soil cores, mixing them up real well, and then pouring that mixed soil into a soil sample bag and sending it to a lab. I kind of belabor this point a little bit, and I don't have a great photograph for it, but I've opened soil bags, and there's like 3 cores in there, and so and they're just still intact cores, and so it makes me wonder, did the person only take 3 cores, Or did they collect 20 cores and only 3 made it into the bag? But obviously, if they take 20 cores and only 3 make it to the bag, those other 17 are wasted effort. So we need 20 cores mixed up really well and then placed into the sample bag and sent to a lab. So when do you sample? Well, I'm a flexible kind of guy. So you could sample when you want to. You could sample in the spring if you're worrying about this and you haven't planted yet. You could sample in the fall after you've harvested and you're scratching your head about why you got 37 bushel beans in a field that should be producing 67. Or if you're on the top of your game and you know soybean cyst is around, you could sample corn stalks in the fall after harvest, getting ready for next year's soybean crop. In fact, that to me is an ideal scenario to sample. It's fall sampling of fields that will have soybeans grown the following year. And then you can even sample during the season. If you're not into digging roots and looking for females on the roots, there's nothing wrong with pulling soil cores from the root zone of a soybean crop in the middle of the season. It really doesn't matter. The next thing is, what should you look to be extracted and counted when you send it to the lab? There are not many labs that offer both of these options, cysts and eggs, but many will offer one or the other, and it really depends somewhat on geography. There are certain states where the culture is that they do cyst counts, and there are other states where the laboratories, both university and private, do egg counts. And so I wanted to just touch on this and make sure you are aware of this, because obviously, going back a slide here, this is a cyst that could contain hundreds of eggs. And so it's counting clusters or clumps of eggs, whereas egg counts, they take the extra step and they break this open to count the individual eggs. So first off, neither one is wrong. Both are fine if you want to test a field to find out whether soybean cyst nematode is there or not. Egg counts give you more precise information than cyst counts, obviously, because you're breaking open those cysts and counting the eggs. When you count the cyst could have, it could be an old cyst that only has 20 eggs left in it, or it could be a new cyst that has 200 eggs in it. And you lose that precision when you do a cyst count compared to an egg count. Cyst counts are cheaper than egg counts because it's one less step. So there's an advantage for cyst counts. And then SCN research is almost universally done with egg counts. And so if you want to read research and apply it to a particular farmer's field, it would be easier to do if you did the egg count. So there's no right or wrong answer. Just be mindful that some labs do cyst counts, some labs do egg counts, and there might be, I guess I should never say never, there could be labs that offer both. Finally, I want to touch on variability of results. I will say that to any agronomist, you will never encounter any situation with data that is more variable than soybean cyst nematode. I'll match my variability to anybody's, and I'll come away a winner or a loser, depending on your perspective, almost all the time. SCN egg counts in particular are extremely variable, and the reason is shown here. So this, I think, is a photograph I showed earlier. It's my hand, my thumb shows my poor cuticle health here, but it shows my finger. It's a normal size finger. That gives you a feel for how big the females are. They're visible with the naked eye. And this red circle is the size of a tip of a soil probe. So imagine if instead of me holding that root was growing in the soil, and we placed the probe in the ground right there, we would have captured 1, 2, 3, 4, 5 females in it. And if I had put that soil probe down a half an inch away, I missed 5 females, full of 200 to 250 eggs each. And that results in this situation. This is a cartoon of what I just showed you. This in panel A shows you the soil probe in the ground collecting 2, 4, 6, 7 cysts, and probe tip B a half an inch away, missing those. And that's not plus or minus 7 objects if you're dealing with egg counts. It's plus or minus 1,500 eggs. And so a half an inch gives you 1,500 egg variability. That's why egg counts are very variable. Just to drive this point home once more and to beat a dead horse, I'm data driven, I'm a researcher, and so this was a research experiment I did many years ago. and it's small plots. Our research plots are 4 rows wide by 20 foot long. So each little rectangle here represents 1/200th of an acre. And we collect a 20-core soil sample from each one of those blocks, 10 feet by 20 feet. And here are the end of season egg counts from plots that were planted with a susceptible soybean. And the amazing thing we see in the top right is 10 feet away we go from 0 eggs to 11,000 eggs. So it's easy to imagine when you think about what I just illustrated in the previous slide. It also shows you some pretty high egg counts that can happen in nature, 23,000, 32,000, 37,000. So the last point I want to make on egg counts is that everybody, it's human nature, you want to know, you get a number back, but how bad is that? Is that low? Is it medium? Is it high? Well, there are several different ways that labs extract soybean cyst nematode, and so their egg counts, the severities really vary depending on the method. And so here's an example of our range of egg counts and how they match to low, medium, and high. If soybeans are going to be the next crop grown, anything below 2,000 I consider low, 2 to 12,000 is medium, more than that it's considered high. And if soybeans are not the next crop grown, in other words, if corn or something else is coming up for a year, we know numbers are going to drop. Those numbers for low, medium, and high are doubled or increased at least. But the big point here is what's in bold red font, and that is that this panel of egg counts is for my method, and that may not be applicable to a method used by another laboratory. And the golden rule is check with your own state's land-grant university, because those folks are the experts and they'll know what methods they're using and what things are low, medium, and high for those particular states. Okay, next thing and final thing is to talk about management. How would you manage soybean cyst nematode? Well, kind of flippantly we could say don't grow soybeans, but none of us want that. Soybeans are an instrumental crop to the agriculture of North America. Soybeans, I live and breathe soybeans. We want to stay in the business of growing soybeans. But the fact is, when you grow a year of a non-host crop, numbers will drop. And so it is an effective management strategy. And we don't want anybody growing continuous soybeans, don't get me wrong. So what happens when you grow a year of corn, at least in Iowa? What happens is the eggs release those juveniles that hatch out. And if you remember, early on in my presentation, I mentioned this is a weak link in the life cycle. This thing, the juvenile, once it hatches, there is no way that it can survive to next year's soybean crop if it hatches in this year's corn crop. And so what we see is 1 year of corn will drop SCN egg numbers from as little as 5 or 10% to as much as 50% in 1 year of corn. And I know a lot of folks that take this and think, well, then I'll just grow 2 or 3 years of corn and really bust it down. Unfortunately, that doesn't work. It's not a linear relationship. Second year corn is not as effective as first year corn at reducing SCN egg numbers. And third year corn is even less effective. And so what we see is a situation illustrated in this particular graph. This is showing end of season egg counts under various crops or after various crops. And so in the orange bar, we see a soybean crop leaving behind, let's just call it 18,000 eggs per 100 cc's of soil. 1 year of corn can drop that down to 9 or 10,000. I've seen it happen in Iowa, and that's wonderful. That drop is amazing. But if you grew a second year of corn, that drop isn't going to be as much. And after about the third year of corn, the curve or the slope of the decrease really starts to flatten out. And what we think is happening is back to biology and the eggs in the cyst. The eggs inside the cyst, we believe, have one of 3 different types of hatching behavior. Some hatch just when it's warm and moist, and so they're going to hatch in a year of corn or sunflower or whatever. But others are inducible, and they're only going to hatch when they sense chemicals given off by soybean roots. And then finally, there are eggs that are going to be dormant. And these are the eggs that Bob Riggs and Darrell Slack have reported that are able to live in soil with no plants being grown for decades. Those are the dormant eggs. And so what we think is most of the constitutive eggs that are hatching just because it's warm and moist enough are going to happen in year 1, and some will hatch in year 2, and there's not much left after a couple years of corn except for eggs that are either inducible and are waiting to sense exudates, chemicals given off by soybean roots, or ones that are dormant. But nonetheless, let's not disregard the benefit of 1 year or 2 years of a non-host crop knocking numbers down. The next management tactic has been a great one. I actually don't think we would still be growing soybeans if it weren't for SCN-resistant soybean varieties. It's literally black and white, or in this photograph, dark green versus pale green. Resistant on the right, susceptible on the left. And the bargain of the century there is that seed of a resistant variety doesn't cost any more than seed of a susceptible variety. It's completely free. This has been a wonderful thing, the free protection we get with SCN-resistant soybeans, but it may turn out to be a fatal flaw that I'll lead us to here in a few slides. So back to the photograph of the swollen juvenile in the root, if you recall from the life cycle. In resistant varieties, the feeding site either doesn't form at all or it dies after a few days. And we know that this nematode can't complete its life without that feeding site. And so the nematodes, juveniles, starve inside the roots of resistant soybeans. The worms still go in, but they're either unable to feed or they feed for a day or two and then they die off. And the time it takes for this dying off of the feeding site to happen can vary a little bit depending on what type of resistance the soybean variety has. But the mechanism is ultimately the same. Worms go in, they try and set up a feeding site, and it doesn't happen. I tell farmers that resistant soybeans pay them twice. The first payment is in yield, and so here are some yield data from resistant versus susceptible soybean varieties. The pale blue bar is the average yield of about 60 resistant varieties grown in a nematode-infested field, and the dark blue purplish bar is 4 susceptible varieties, widely grown susceptible varieties. And in a typical field in Iowa, we could see these types of yields, 46 bushels versus almost 53 bushels. So that's 7 bushels per acre, money in the bank profit. That's the first payment. But there's a second payment that'll come due, or they'll receive, the farmer will receive a couple years down the line, and that's from the nematode control. So in these 2 bars to the right in the graph, the pale bar is the end of season egg count left behind under the resistant soybeans that yielded 7 bushels more, and the purplish blue bar is the egg count left behind under the susceptible. And remember, we started out at 1,300 eggs in this particular field. 1,300 versus 2,100, that's almost a wash when you consider the variability in egg counts. But clearly, egg counts quadrupled, increased 5-6-fold underneath those susceptible soybeans, which gave the farmer 7 bushels per acre less. So these higher egg counts are going to ding soybean yields the next time soybeans are grown, and the lack of eggs left behind under the resistant is the second payment. So almost from the first year that I worked at Iowa State University, I started putting together a list of cyst-resistant soybeans. And I've made a bar graph here of just the number of varieties available for Iowa farmers from 1991 up until 2016. We're about to release our 2017 list here in a week or two, but I haven't counted them yet. But it's wonderful. This was 29 varieties in 1991, and this was 956 varieties in 2016 that Iowa farmers could pick from. So it's been a great success story. A couple points about the biology of resistance, SCN resistance. It requires several genes, and so it's not a single gene resistance like Phytophthora is, for example. It involves several genes. They're called RHG genes, and RHG stands for resistance to Heterodera glycines, which is the scientific name for soybean cyst nematode. And then recently, in a paper published just a few years ago, scientists have discovered not only was it the presence of genes, but also the number of copies of a set of genes that determined how resistant a variety was. You may or may not be surprised to know that there's no legal definition of a resistant soybean variety in the United States. There is some standards used in Canada, but not in the United States. But there's a scientific definition that we all kind of abide by, and that is that a resistant variety should allow less than 10% reproduction relative to a susceptible variety. In other words, there should be 90% suppression or control. And if you're into scientific literature, the very bottom citation there, this is a journal available online, Phytopathology, and Melissa Mitchum from University of Missouri did a really nice review of the state of resistance if you wanted more information. Well, when you start looking at descriptions of resistant soybeans, you very quickly encounter something called sources of resistance. And these are breeding lines that soybean breeders use to introduce resistance genes into really good agronomic soybean varieties. So imagine you have a great variety that yields 75 bushels per acre in non-nematode fields, and you want to make it resistant to SCN. You have to cross it with one of these breeding lines. And there are 7 main ones that breeders consider, but there are hundreds of others that plant geneticists and breeders have identified as well. And so they literally cross. They take the pollen from one plant and put it in the flower of the other, and then test the offspring to see both, did they retain the high yield characteristics that they want to keep, and then introduce the resistance genes. And so if you remember my bar graph of the number of varieties in Iowa from 1991 to currently, the next slide I'm going to show you is the distribution of the types of resistance or the sources of resistance used. And it's a real simple bar that has mainly one color. And that one color is gray that matches up with the one breeding line PI 88788 for resistance. Everything else, the other 6 common sources of resistance plus the 100 or more others that are known, very, very few varieties are brought to market with those resistance sources. It has something to do with the yieldability. When you transfer those resistance genes into a good agronomic bean, somehow there's yield drag that's associated with it, and it's hard to get both good resistance and high agronomics. And as a result, 97% of the soybean varieties that an Iowa farmer could pick from to grow have this same source of resistance. So I ask you, can you imagine what would happen if we used a single herbicide active ingredient for 20 years, over and over and over? Can you imagine? Sure you can. And the exact same thing happens with resistance genes, and we're starting to see it. Oh, and to complicate things a little more, very few soybean varieties brought to market these days don't have PI 88788 resistance. It's an automatic in the background. All the breeding, all the varieties that soybean breeding programs use to improve upon already have the 88788 resistance. So there is a test that people like me do on farmers' nematodes to see how well they reproduce on these main sources of resistance. So I can work with a farmer. He or she will collect a 2-gallon soil sample. We need a lot more soil. And we'll grow all these different sources of resistance in that soil to see what that farmer's nematode will do on these different sources of resistance. It's called the HG type test. And lots of people are doing that to get a feel for what's going on out there in the countryside. So here are just 6 or 7 publications, and I put the states that are represented from these publications of people that have done HG type surveys. And what they're doing is they're surveying their fields, fields in their state for nematodes, and then they're testing them on these 7 sources of resistance. And so again, here are the citations from Ontario and South Dakota all the way south. And in summary, without going into excruciating detail, SCN populations with greater than 10% reproduction, it's called a female index, is common now in most areas. And so remember, 10% was the standard definition of resistance. PI 88788 provided resistance, but now the nematode is overcoming it because our reproduction levels we're seeing are 50 and 60% on PI 88788 now. And then there have been a couple of these published surveys that they had done like 10 years ago, and so they can compare what the results were from 10 years prior and 10 years now. And they see greatly increased reproduction. And the most frightening thing is the bottom bullet point here is there's no way to go back. We can't undo this buildup of the ability of farmers' nematodes to feed on 88788 resistance. Now, I never did one of those surveys, but I've evaluated resistant soybean varieties for decades. And so I was feeling a little self-conscious that I didn't have survey results to show. But someone pointed out that when we do our variety trial experiments, we do it at 9 locations every year in Iowa in farmers' fields. And first thing we do is we take a large soil sample from the farmer's field and we test the farmer's nematode on pure 88788 in Peking. So I have 25 years of data of nematodes in the state of Iowa in fields and how well or how well not they reproduced on 88788. So my variety trial data is kind of a survey of reproduction of SCN on 88788 in the state of Iowa. So we look back at 25 years of data recently, and in the decade of the 90s, almost all SCN populations in our farmers' cooperators' fields were well controlled by 88788. Reproduction was below 10%. But here's a graph of what happened starting in the year 2000. So let me orient you here. This is reproduction of the farmer's nematode on pure 88788. Each data point represents a variety trial field. So these are farmers' fields that we sampled and then we grew pure PI 88788. And remember, in the 90s and even in the year 2000, reproduction was below 10%. That's what we want. And I've drawn a green line here to show that's where we want to be below. But as the years have progressed, you're seeing the percentages in terms of these dots. And so this illustrates in farmers' fields in Iowa that we've done variety trials, the gradual buildup of their nematode on 88788. And the farmer manages the field however he or she wants to. We just rent a chunk of their land every year to do our variety trials. We never go to the same field or area twice. It's always new and it's always following corn. So we believe this is representative of what's happening in Iowa. Now the real neat thing about this is we did variety trial experiments in there. So we have yield data of resistant soybeans in these fields. And here is what we see. We also have nematode reproduction counts. And so here is the same years that I showed on the x-axis as in the previous slide. And this is the reproductive factor, or what we call RF, which is basically the end of season number of eggs divided by the beginning of season number of eggs. So if you had an RF value of 2, it means your egg counts doubled, because the final number would be twice as high as the beginning number. An RF of 4 would mean the egg counts quadrupled. So here are the RF values, and no, your eyes aren't deceiving you. There are values of 10, 15, 20, 25, one crazy one of 500-fold increase. This is the reproduction in terms of egg counts left behind under varieties with 88788 resistance in those variety trials. And then, as I mentioned earlier, we also have yield of those varieties plotted against the reproductive factor. So now RF, or reproductive factor, is on the x-axis. Yield of those Peking or 88788 varieties is here, and we see a gradual decrease. This decrease in yield as nematode reproduction increases makes sense. As nematodes reproduce, it's causing harm and yields decrease. It doesn't look like a dramatic decreasing slope, but there's a 14 bushel per acre difference in yield of resistant beans from the end of the line near 0 and the other end of the line where RFs are at 30 or 40. So clearly our resistant soybean varieties are losing yield. So here's a summary slide of my situation. And I don't believe it's probably much different from many of your situations, at least in the Midwest. We have almost all varieties having 88788 resistance. We have farmers' nematodes built up the ability to reproduce on 88788 resistance. We're seeing egg counts increase under resistant varieties grown in these fields, and we see the yields of those resistant varieties dropping because of the increased reproduction. So it's in many ways a cliche I use as a train wreck in slow motion. So prospects for the future, the usefulness of 88788 is going to continue to decline. I don't see it stopping or changing. And so the days of this kind of difference happening, I think, are numbered. I don't think we're going to see differences like this, resistant versus susceptible, much more. And in fact, I wonder if at some point 88788 is not going to provide resistance to soybean cyst nematode. Not because it doesn't have the resistance genes, but because of the nematode has overcome the resistance. And I'm not optimistic that we're going to see a lot of new varieties with anything other than 88788 resistance. And the reason is there's no money to be gained. So it would take a lot of effort by companies to develop more varieties with this other types of resistance, and they've never charged a premium for it, and so how would that effort be recovered? So we're kind of in a pickle. Earlier I said, this was wonderful that we were getting free protection for decades with SCN resistant. There was never a premium. But now we're stuck because it's going to take extra money and time and labor to produce something different, and it's very unlikely that they'd be able to recover that in an increased price. So the third and final main management strategy is the newest, and that is seed treatments, nematode-protectant seed treatments. And I just want to touch on these one by one. There are 7 of them at the moment. We could be standing here a year from now and there could be 9 or 10 or 11. So it's really cool that the activity going on here. The product that kind of defined the market was something called Avicta from Syngenta, and Cliff was involved in that. And it was a product that was marketed on cotton, I believe, first, and then migrated to corn and soybean. And because of the active ingredient, which inhibited nematode nerve transmission, it had activity against all plant parasitic nematodes. That's what PPN stands for. And around that same time, there was a company out of Pittsburgh, Pennsylvania, Plant Healthcare Incorporated, selling a seed treatment called Inhibit that was a protein that stimulated natural plant defenses. And those natural plant defenses were broad-based. And so it was described as providing protection against all plant parasitic nematodes and could be used on all crops. The third entry into this area was a product from Bayer Crop Science called Votivo, again available on cotton, corn, and soybeans, having activity against all plant parasitic nematodes. And this one was a biological, a bacterium, Bacillus firmus, that didn't kill nematodes, but protected the roots by repelling nematodes from the roots. Syngenta did a follow-up to Avicta with a newer product, maybe 10 years later, 5-10 years after Avicta, called Clariva PN, and it's strictly for soybean because it's very specific for soybean cyst nematode. And it's another biological, but this is really interesting because it's a parasite. It actually is a bacterium that infects soybean cyst nematode juveniles and kills them. After that, Bayer released a product mainly targeted for sudden death syndrome. It's a fungicide seed treatment called Alivo, but it was found out that it had activity against soybean cyst nematode, root knot nematode, reniform nematode, and lesion nematode. And the amazing thing there is it does look like the fungicide active ingredient, which is an SDHI enzyme inhibitor, is the component that's also affecting the nematode. The most recent one, for today at least, in the fall of 2017, is Monsanto's Nemastrike, which is available for cotton, corn, and soybean. The same suite of nematodes as Alivo. It's a compound tioxaphene. I'm still learning how to pronounce that. And it's a mitochondrial translation inhibitor. And also coming very soon, I believe it'll be registered and available next year, is Aveo. It's a product coming from for corn and soybean with activity on soybean cyst nematode, reniform, and lesion. And it's another biological. It's actually not going to be the bacterium as the seed treatment, but a product of the bacterium. And the mode of action has yet to be described. Some final thoughts on nematode-protectant seed treatments. I'm not sure everybody has thought about this as much as I have, but going into it, I thought a farmer could just buy a Clariva or an Olivo, but everything is pretty much bundled with base, seed treatment, insecticides, and fungicides. The one exception would be the Inhibit product, and the Inhibit product is one that farmers could buy directly and apply themselves. Also, another thing is I would think that if we would buy seed from Pioneer, you could pick 3, 4, or 5 different seed treatments to pick from, but that's not true either. In today's business world, all the large companies have aligned themselves with pretty much one lineup of nematode-protectant seed treatments. So if you want a particular seed treatment, that's going to steer you, at least in the large companies, towards one seed company's lineup. These nematode-protectant seed treatments can increase yields for sure, but the results may vary among fields and among growing seasons. And SCN population densities will build back up as that protection wears off. But a silver lining to all this, coming off of my comments on the slow erosion of PI 88788 resistance, we sure hope that these seed treatments slow down the gradual demise of 88788 resistance. And the final thing, which is really enlightening or uplifting to me, is we just don't know what's out there that's going to come to market next. And so every product seems to have a new mode of action, a new mechanism, and so it's hard to even guess what's going to be next. I guess one last final point I'll make. I'm a university researcher, and we use small plot equipment to do small plot experiments, and it may be easier, especially for yields, to get significant differences in strip trials rather than small plot experiments. And then the other thing is we're doing all the testing with resistant soybeans, and it might be easier to pick up treatment differences with susceptible, but they really are meant to be sold on resistant seed, so that's the more logical way to test it. So this is how Greg Tylka views seed treatments for nematode protection. We have SCN control on one axis and yield increase on the other, and there's 4 possibilities shown here, and the worst place to end up at the end of the season is having received no yield increase and no nematode control. What you'd hope for is a yield increase. That's probably more important to a farmer than nematode control. To Greg Tylka, I'd like to see a reduced soybean cyst nematode, but it can happen without a yield increase, and the top right obviously is where we want to be consistently, and that's what we're working towards. This is my final slide. This is the future here. Non-host crops, we want people to grow non-host crops because it lowers numbers. We got to keep growing resistant soybeans. It hurts me to say, even those with 88788. I mean, we don't have a choice. We have to. Farmers are going to need to seek out varieties with Peking and other sources of resistance, and if something new comes on the market, boy, I think we're going to have to really work and adapt and adopt those as well. We need to consider using these nematode-protectant seed treatments, and then I'm getting lots of questions about cover crops and if they could play a role in managing soybean cyst nematode. To this point, there are glimmers of promise that some crops, under some conditions, cover crops can reduce SCN numbers more than if there was no cover crop grown, but the consistency has yet to be determined. Thanks.